In this video, I'm going to explain the process of cellular respiration. And the first thing that you should recognize with cellular respiration is that the reactants of cellular respiration, which are oxygen and glucose, those are actually the products of photosynthesis. And the products of cellular respiration, which are carbon dioxide and water, those are the reactants of photosynthesis. So these two reactions are actually exact opposites of each other. Now, there are a lot of similarities between photosynthesis and cellular respiration, and during this video, I'll note them. The one difference, though, just to start off with differences, is that there are 3 steps to cellular respiration while there are only 2 for photosynthesis. So I'm going to go through and I'm going to explain these 3 steps. The first step is glycolysis. Now, what happens in glycolysis is that glucose, which is a 6-carbon molecule, is broken down into 2 pyruvic acids, which are 3-carbon molecules. Now, other than the fact that glucose is being broken down into pyruvic acid, there are 2 other things that are happening. First, in order to get this reaction going, ATP is required. So it uses 2 ATPs and makes 2 ADPs. Now, the interesting thing is that in the end of glycolysis, 4 ATPs are going to be made. And so what that tells us, since I put in 2 but I get 4 in the end, that means I have a net of 2 ATPs. And the other thing that's being produced, I have NAD+, which you should recognize as being super similar to NADP+. That's the same molecule, slightly different, that's used in photosynthesis, and it's an electron carrier. Now, NAD+ grabs electrons as glucose is being broken down into pyruvic acid, and it produces NADH. Now, this NADH is going to be then shuttled off to the electron transport chain, and we'll see how it's used later on. Okay, so glycolysis, glucose is made into pyruvic acid. You get a little bit of ATP, only 2 molecules of ATP, and then you also produce NADH. The next step in cellular respiration is the Kreb cycle. And the Kreb cycle, pyruvic acid, which was made in glycolysis, which again occurs in the cytoplasm, that pyruvic acid moves from the cytoplasm into the matrix of the mitochondria. Now, in this process, the carbons of the pyruvic acid are slowly and further broken down into carbon dioxide. So we're getting rid of the carbon, which makes you think, okay, well, then what's the purpose of the Krebs cycle? Well, the purpose of the Krebs cycle is to produce not only extra energy, ATP, it's not that much though, but also NADH, which again is my electron carrier, and a brand new electron carrier called FADH2. FADH2 without electrons is called FAD. Now, FAD picks up 2 electrons and 2 hydrogens to become FADH2. So FADH2 and NADH will then be shuttled off to the electron transport chain in order to create more ATPs, which is the ultimate goal of cellular respiration, is to create usable energy in the form of ATP. So that brings us to the third step. So the FADH2 that's made just in the Krebs cycle, and the NADH, which is produced in the Krebs cycle and in glycolysis, as we saw above here, are then going to be shuttled to the electron transport chain. The electron transport chain is super similar to the electron transport chain in photosynthesis. Instead of being in the thylakoid membrane and having the thylakoid space in the stroma, which you should go back and review again, what we have here is the matrix of the mitochondria and the intermembrane space. And so the electron transport chain is in this inner membrane right here, but it looks super similar to the electron transport chain in photosynthesis, which it is. Okay, so NADH and FADH2 are going to be delivering the electrons this time, okay, which is kind of ironic because in photosynthesis, water was what delivered the electrons. But the electrons move through just like in photosynthesis, and then they're going to be picked up. Now, again, ironically, the electrons are not picked up by NAD+ or NADP+ like it was in photosynthesis, but instead it's picked up by oxygen, which we call our final electron acceptor, and with hydrogen it produces water. Now, the reason why I said this was ironic, remember, in photosynthesis, water was what provided the electrons, and NADP+ was what picked them up at the end of the electron transport chain. So now it's completely flipped. Now we have NADH delivering the electrons in addition to FADH2 and water being produced as a result. So these are really completely opposite processes. The other concept that you need to remember is the fact that as the electrons move through the electron transport chain, they power the hydrogen pumps, which pump hydrogen from the matrix into the intermembrane space. So you've now got a high concentration of H+ in here. Now, in photosynthesis, the other source of those hydrogen ions was water. But what you need to remember here is we're not in photosynthesis. So instead, what happens is in the matrix, since water is not being broken, it's being formed, the hydrogen ions are actually being removed from the matrix, right, because they become water. And as a result, this causes the hydrogen concentration in the matrix to get even lower. So we're not adding more hydrogens here other than what the pumps do. We're actually just removing hydrogens from the matrix. And so as a result, you have a super high concentration of hydrogens here and a very low concentration of hydrogens in the matrix. And so as a result, this is going to power the ATP synthase. Now, the ATP synthase is powered because hydrogen ions want to move from high concentration to low concentration, and this movement powers the ATP synthase. So it's moving from the hydrogen ions are moving from the inner membrane space into the matrix, which powers the ATP synthase, which makes ATP. So remember, just to compare and contrast with photosynthesis, if you remember, hydrogens moved from the thylakoid space into the stroma, so it was like it was moving out, which powered the ATP synthase, while here it's moving from the intermembrane space into the matrix. So tiny bit different, but still the exact same concepts, which makes it very nice and cool because you're able to compare, contrast, and then it helps you to remember. Now, those are the 3 steps of typical cellular respiration, but what your question should be is, well, what happens if I don't have this final electron acceptor? And so what I'm going to do is I'm going to walk you through it to understand what's going on. Well, if I don't have the final electron acceptor, electrons will back up, NADH won't be able to deliver its electrons to then become NAD+ again, and FADH2 again won't be able to become FAD, which is a big problem because if we go back up here, glycolysis requires NAD+, and the Kreb cycle requires NAD+ and FAD. So if these guys aren't able to deliver their electrons to become NAD+ and FAD, then those 2 other processes can't happen. And so as a result, if oxygen is not present, what cells have evolved a mechanism for dealing with this issue. And so what they do is a process called fermentation. Now, in fermentation, there are 2 different types, but the ultimate goal of fermentation is to reproduce NAD+. And the reason why is NAD+, once you have NAD+, then you can go back and complete glycolysis. Okay, so if no oxygen is present, then the Kreb cycle and the electron transport chain are skipped, but glycolysis is still possible. And remember, glycolysis makes 2 ATP, so that suffices. It's not as good as having the electron transport chain, but that's fine. So the 2 different types of fermentation include alcoholic and lactic acid. So what you should notice, this is glycolysis here. Okay, if I have alcoholic fermentation, then that pyruvic acid is broken down into carbon dioxide and ethyl alcohol. The purpose of this fermentation is to take NADH and convert it back into NAD+. Same thing with lactic acid fermentation. Pyruvic acid is broken into lactic acid, but the whole point of it is to take NADH and remove the 2 electrons to get NAD+ again. Because once that has been created, it can cycle back and you can have glycolysis occur. So fermentation is a way of dealing with the fact that I can't collect electrons have backed up, and therefore NADH is stuck as NADH because it can't get rid of the electrons to become NAD+. Okay? Now, the last thing is if we are running, there are a couple of different processes that occur. First of all, if we just run for a couple of seconds, our bodies use the ATP that's already in our muscles. However, what you should know is that the ATP is not a good way of storing energy, and therefore our body stores it in larger molecules and then breaks it down through a catabolic reaction in order to produce usable ATP. So, two different ways of dealing with exercise. The first way, if we're sprinting all of a sudden, which, as you know, you're not able to get a lot of oxygen during that time, what our bodies do is a process called lactic acid fermentation right here. Okay, so without oxygen, we can still produce ATP because we can still have this cycle. However, it's not that much ATP. And what happens is after about 90 seconds, we have a lot of lactic acid built up, and then our bodies can't keep that going because there's a lot of that lactic acid that is built up. And so as a result, we have to go back to cellular respiration, which means you can't be sprinting for dear life for more than about 90 seconds. Because after that point, you revert to cellular respiration. Okay. The other important thing for you to know is that with lactic acid fermentation, since lactic acid builds up, your body wants to get rid of it. Well, ironically, the way to get rid of lactic acid, to break it down, is to get oxygen. Oxygen will break down lactic acid. So after you've been sprinting for a while, if you've ever had like terrible cramps and you're sprinting, what you end up doing is panting a lot in order to get more oxygen, to break down the lactic acid, and to allow cellular respiration to occur. Now, cellular respiration doesn't occur when you're sprinting because you don't have enough oxygen and it's not fast enough. Cellular respiration is it takes a lot more time than fermentation, but as you can see, cellular respiration makes more ATP than fermentation does.